In this PPT, I was going to say about PCR, polymerase chain reaction. Introduction: PCR is a molecular biology technique used to amplify the DNA at the specific region, and it also used to create a million to billions of copies, and it was first developed by the Kary Mullis. Principle of PCR: PCR uses the heat stable DNA polymerase, and it also uses the primers. Primers are nothing but the short single stranded DNA sequences, and this steps involves the different temperatures. Key components used in the PCR: NA templates. NA templates are nothing but the sample that containing the specific region of the DNA, and primers. We use both the forward and reverse primers. And DNTPs, it was an nucleotide blocks ATGC, which will helps for the synthesis of new DNA. And the DNA polymerase, we will use the Taq polymerase, which will help the strand to withstand in the high temperature, and we will use the buffer, which will provide the correct chemical environment for the reaction, and thermal cycle, the instrument we used for this process. Steps in PCR: first step is denaturation. In this step, the double stranded DNA was separated into two single strand DNA at the temperature 94 to 95 degrees Celsius. And second step is annealing. In this step, the primers are binded at the temperature 50 to 65 degrees Celsius. In the third step, extension, the temperature was raised for about 72 degrees Celsius. In this step, DNA polymerase will added to withstand the DNA at the high temperature, and the primers will add the new nucleotides at the 3' end for the new synthesis of the DNA, and these steps are repeated for about 20 to 40 cycles. By theoretically, we can represent as 2n. N represents number of cycles. In the both diagram, the first step, double stranded DNA was separated into two single strand at the temperature 94 to 95 degrees Celsius. In the second step, annealing, the primers are added, which will help for the new synthesis of DNA. In the third step, extension, the temperature was raised to 72 degrees Celsius, and the new strand is formed. Types of PCR: qPCR, which will amplify the DNA, and we can view it through the fluorescent dyes. And second one is RT-PCR, which will amplify the RNA by using the RNA. Multiplex PCR amplifies multiple target sequences in a single reaction. And downtouch PCR or overlap extension PCR, it is an specialized model to improve the sequences. Application of PCR: PCR was most widely used in genetic testing like mutations and pathogenetic deductions from very small amount of sample. In very small amount of sample, forensic DNA and research purpose also we can use PCR. Thank you.